Okay, so investing in innovation means that we all try and can showcase a whole wonderful package of things. So included in that, we're actually bringing in a presenter from on the line. So while that's being loaded, that presenter today is Ben Hayes, who is the Centre Director for Animal Science with QAFI and UQ. Ben, you can hear us?
Yes, I can, loud and clear.
Don't you love when technology works? Okay, so Ben
great.
obviously has an extensive research experience in genetic improvement of livestock, crop pastures and aquaculture, with a focus on genomic information in the breeding programs. He has authored over 250 journal papers and is a Thomson Reuters highly cited researcher in 15, 16, 17 and 18. He's here today to talk to us about predicting age of livestock from DNA samples. So Ben, over to you.
Yeah, thanks very much. And thanks to the organisers for inviting me. I'm very sorry I couldn't be down there with you. Just to pick up on Harry's presentation, I had a COVID test a few months ago, and they used the nanopore technology to check whether I had COVID or not. And I had the test back in 45 minutes. So it is a pretty rapid turnaround. And as Julia said, that's what's really driving that technology. And we're just riding off the back of that, really, and adapting that over to livestock. So that's where the big cost cutting is going to happen. So I'll launch into quite a different problem, but come round to a little bit of a similar place. And the problem we're going to tackle today in this presentation is predicting age of livestock from DNA samples. Okay, so this is Harry up in the chopper mustering some cattle. Don't think so, actually. I don't think Harry can actually fly a chopper. But anyway, it just brings home on these northern cattle properties how extensive these operations are and how expensive it is when you want to bring the animals in for sampling, data recording, and so on. And one big problem that leads to is it becomes quite difficult to know the age of your animals with any precision at all. So if you muster once a year, it can be quite hard to know, is this animal born last year or even the year before or early this year, and so on. You just have no read on how old that animal is. And that causes a variety of problems. One is if you're putting these animals through the works and you don't know how old they are, it's hard to get MSA, for example, good quality meat gradings. It also has the problem, if you want to do genetic improvement and you do want to record animals, if you don't have that birth date, you're in trouble, because that's where the whole genetic evaluation, genetic recording really starts, because age is such a key factor in the models we fit to various traits like growth and fertility and so on, or can be such a key factor. So is there any way of telling how old these animals are from a biological sample, from a sample taken from the animals? And ideally, we don't want to completely reinvent the wheel here. We want to use the same sample that's used for other things, and particularly for these days for genotyping. So either tail hair or an ear punch. I think in sheep, TSUs are very widely used now. So there you'd want to use a TSU. In cattle, we still use a bit of tail hair as well. So can we work out the age of animals from a tail hair sample or an ear punch sample? From the DNA, this initially sounds like complete fantasy, because as I was told in undergraduate courses and so on. And as I'm sure people have told you, the DNA is 4 letters, A, C, T, and G, and that's it. And it's stable through the animal's life. And that's incidentally why it's so good for genomics, because it is stable. You can make a prediction from when the animal's just born, and the genetics, the DNA, will be the same throughout the animal's life. But it turns out there is a subtle twist to this. As we get older and older, our DNA gets something added to it called methyl groups. So this is just a molecule that gets added to it. And the reason these methyl groups get added to the DNA is to start switching genes off. So that's one reason that as we age, things start to go a bit pear-shaped and deteriorate, because some of our genes for repair are just being switched off. Now, these genes can be switched off in response to environment. So if you starve an animal or starve yourself, some of your genes for metabolism might be switched off. It turns out some genes are just switched off with age as well. So they just get, every year, more and more genes get switched off, or certain genes get switched off. And that process is very, or appears to be, from the experiments that have been done, very repeatable. This just happens month after month, year after year. So the researchers that realised this had the inspired idea, I think, that this can be used as a methylation, or you can also call it an epigenetic clock. This process of switching genes off, this is a part of epigenetics. So this has been taken up in quite a big way in humans. And what people have done is done methylation profiles of people. So looked at how many genes are switched on and off based on these bits that are attached to the DNA, and then just correlated that or lined that up with people's ages. And it turns out you can predict someone's age reasonably accurately with these methylation clocks. So the graph on this slide shows the relationship between actual age, which is on the x-axis, going up to 70 years of age, and the predicted age of one of the first methylation clocks that was developed. And you can see it's not perfect, but it's pretty good. And in fact, for humans that live about 70 years, you can predict the age of a person from a hair sample or any sample containing DNA, really, within plus or minus 4 years. You can imagine the forensic community has gone pretty nuts over this, because they can go to a crime scene, pick up a hair, and work out how old the criminal was, and then do profiling on that person. This idea of the epigenetic clock has been rolled out across a number of different species, including mice. Mice live just a few months, and in mice, the accuracy of the clock is really good, reflecting that shorter lifespan. There's less variation there, so you can predict age within plus or minus 2 weeks. Dogs are quite interesting as well. There's this old wives' tale or paradigm that every dog year is worth 7 human years, so dogs age a lot faster than humans. It turns out when you look at the epigenetic clock for dogs and humans, that's not quite true. Dogs actually age quite a bit faster as puppies in the early stage of life, and then their aging slows down in later years, whereas humans just keep ticking along at a fairly steady rate. So that's interesting. But more interesting for us is these epigenetic clocks or methylation clocks are a potential way of aging our livestock. When we don't have the ability to record birth dates, or when it's dangerous to do so because you have animals with undesirable temperament and so on. So the first thing we did in this project was ask the question, is this likely to be cost effective, and what technologies are likely to be cost effective? There's a whole bunch of technologies that will give you this methylation profiles on the DNA, so it can be used as the data for the methylation clock, but which ones are really going to be useful? And we found out fairly early on that the nanopore technology using these mini iron devices, so this is the sequencer here, can also be used to get these methylation profiles. As Harry was describing, in these devices, the DNA gets passed down a little tube, a nanopore, and the DNA sequence gets read off based on these the current going across it. Turns out if your DNA is methylated, has these methylation groups attached, then the current is slightly different. So you can turn this output from the nanopore, from the little sequencer, into a methylation profile. And then hopefully you can predict the age of an animal from that. What you need though, just like in genomic selection, is you need a reference population to work out, is a particular point on the genome that gets this methylation, which is switching the gene on and off, is that associated with aging, or is it just reflecting the environment or something else? So can we use it in our clock or not? It looks like from all the other studies that have been done in other species, you can get away with a moderate sized reference population because the methylation signals are much bigger than what you get from SNP chips for the traits that we're interested in. So you might only need a few methylation spots, several hundred, to get one of these methylation clocks that is quite accurate. And in dogs, for example, they used 80 samples. We've decided to use 100 samples in our clock, and we deliberately did that across different herds so we could sort out different environments from the age effects on the profiles. And we also had a span of years within the herds. And we also took samples from animals when they were calves and when they were mature cows. So we had an internal control, same animal, but what does its methylation profile look like at different ages? So all up, we've got 100 cattle that have been sequenced with this technology. And so the sequencing has been completed, the methylation profiles have been done, and now the last part of the work is to derive that clock, which we're doing right at this moment. So I apologise, I don't actually have the clock to present to you. That's still in progress, but all the sequencing work and the preliminary analysis has been done. What we do see, this is just the age range in the different herds. We deliberately chose some herds with the great span of age. So the first herd covers 2 to 10 years. Other herds, we wanted to look at getting more precise estimates. So can we tell the difference between just a few months? So we've got cattle in there where their age is spread across just a few months. And I should say these are breed plan herds where we have very accurate age at birth or dates of birth. So we can work out age pretty precisely for our reference population. When you go ahead and sequence the animals, what you see is huge variation in the amount of the genome that is methylated, so where the genes have been switched off. On the x-axis here, you just have the sample number, the animal number. On the y-axis, you have how many points along the whole genome have been methylated and switched off from genes. And the largest ones have 500,000 or more spots where the genome's been methylated. The smallest one has a lot less. Some of this is sequencing coverage, but some of it also reflects the age of the animal, how many genes are switched on and off, and some of it reflects the harshness of the environment as well. What we have already done is analysis of a small number of samples, and we can very clearly predict year groups. So from the methylation profile within the same herd and across a couple of different herds, we can very easily see from the methylation profiles whether they were, say, 2015 born animals or 2016. So telling apart a year of birth is pretty straightforward, even from this small sample. When we process our large sample and analyse it with 100 animals, we should be much more in the region of a month or weeks of birth. So that brings me to concluding this talk. Epigenetic clocks have been developed in humans and dogs. The accuracy's a couple of years in humans, 4 years. In mice, it's a lot shorter, and it just depends on the lifespan in the animal. In cattle, given the lifespan of 10 to 12 years, we're hoping we can get down to plus or minus a month or even better. And that would be as good as some of the recording that goes on for birth dates that are currently used. So once we have this up and running, and we're going to combine it, of course, with Harry's crush side genotyping, so you get back both genomic breeding values and age of animal, and enabling your records to go into breed plan as well. We really hope this leads to more widespread adoption of herd recording in the north, or these challenging extensive environments, and greater genetic gains. And I'll just finish up by saying, I won't expand on it, but you can predict from these clocks both chronological age, so age in years and months. You can also predict biological age. So some of us that have lived harder lives than others, our actual biological age, the age of our bodies, will be older than our chronological age. Some of us that have had cushy lives might have much younger. Our chronological age might be 70, but the age of our bodies might actually be 60 years. And you can also predict that. So we're looking at that. Can we predict ossification, for example, for Meat Standards Australia? Okay, I'd like to thank very much the head of our lab, Elizabeth Ross, Harry and Lone, and the collaborating breeders on this project. And of course, MLA. I just realised I haven't got the MLA logo there. I very much should have, because they funded this project. So thanks very much.
Thanks, Ben. So we have 2 questions online, Ben, and I'm just going to, we'll take one from the room. Obviously your data set is based on cattle. Would you expect something similar in sheep?
Yeah, absolutely. I think because this has been done across a range of species, there's no reason you couldn't do it in sheep. You just have to design your reference population appropriate to sheep lifespans, and so that you were predicting maybe it's weeks there that you could get the birth dates within. Excellent.
So I'll follow on from that, Ben, with our second question. Around, obviously, age prediction, and you spoke about biological clocks versus physical clocks in terms of the value of selecting animals that might age slower or faster.
Yeah, so it seems like part of the methylation is just age year after year, but as I said, part of the methylation is environmental. So if animals go through a drought, you should clearly be able to see in their methylation profile that they've had a hard time. And the beauty of that would be you could put that into your genetic evaluation. So the growth rate of this animal, for example, or the intramuscular fat is lower than you expect given the genetics because it's been through a tough time. And you can pick that up through the methylation pattern. The best example of this I know of is smoking. So if any of you out there are smokers, your genome is pretty highly methylated. You can just see as clear as day in the methylation profile whether someone smokes or not. That ages you much more quickly than your chronology would predict, than your age of birth and so on would predict.
I think there'd also be a relationship there. I know my parents probably have aged horribly as a result of having to look after me from a young age as well.
Yeah, that would definitely do it.
Yeah, we've got a question
Like
here
it.
in the room.
Okay. Actually, Lena from Agbu.
Could you just hold the microphone a bit closer? Yep.
Okay, now you can hear me. Yeah. Okay, Lena from Agbu. And actually, I have a question. Maybe it's somehow common. I don't know. Actually, I just wondering that, as you know, that methylation, methylation of DNA associates with some disease. So I just wondering that, have you ever put that in your model to actually as effect to adjust it for that? I suppose that is the reason that you have plus minus 10 years actually that deviate for your prediction, I mean.
Yes. Yep. No, very good point. We haven't done it yet, but in humans, that's where this whole field of epigenetic clocks is going. So they're looking at if you have unfortunate enough to have cancer, how does that biologically age you through methylation and so on?
Yeah.
Which genes is it actually switching off? If you have a heart attack or heart disease, how is that reflected in your methylation pattern? And indeed, can you use that as a potentially a predictor if it's a chronic disease? Can you pick it up early from a methylation profile? So there's a lot of good work going on in that field in humans, and that's where this technology is going now. So good question.
Yeah, 3 very good questions there. I'm going to pull it up, Ben, if that's okay. Thank you for dialing in today. Good to see the fan going in the background, meaning it's another typical beautiful Queensland day. But ladies and gentlemen, just give it up for Ben one more time.